All right, bioinformatic detectives, here is another edition of how to find the culprit. So in this exercise, we have a sequence, a nucleotide sequence that was sequenced from an important drug-resistant pathogen. We want to find out what the pathogen is and what does the gene do. In this information we have is that this is a mRNA messenger RNA sequence, and we want to know what the protein does. So we are using today BLASTX because we start with a nucleotide sequence and we want to know what the protein does. So let's go to BLASTX and copy and paste our sequence. Again, we can see it's a nucleotide sequence, and sometimes in terms of formatting you see that there are numbers in there, but BLAST is pretty forgiving with numbers and formatting. Some other programs you have to change the formatting, get rid of the numbers, and I will show you this in another video. So this is our nucleotide sequence of a drug-resistant pathogen, and we want to know what pathogen it is. And again, we're using BLASTX. Alright, so let's do the BLAST and let's wait. Excitement is mounting because we don't know what the organism is. We just have this gene and we are gene detectives. We're working for the CDC and they are on their toes to figure out what this pathogen is. Give it a few more seconds. Alright, let's see what our results are. What is the mRNA sequence in this pathogen that's really important? Alright, so let's scroll down on our BLAST results, and it is full beta-lactam inducible penicillin binding protein, Staphylococcus aureus. Sounds pretty mysterious. Let's do a little bit more research. So Staph aureus is definitely potentially a pathogen, MAC-A protein, so it gives us some information here. We're still not quite sure what it does. It does sort of beta-lactam inducible penicillin binding protein. So just from the name we could deduce that this resistance protein binds penicillin so that the pathogen does not get killed by the antibiotic. I guess that's probably what it is, but let's do some more research here. Alright, there's a couple of papers here. Keywords: antibiotic resistance, cell cycle, cell membrane, and so on. So lots of good information. And this paper probably would be quite interesting to look at. Evolution of inducible penicillin targeted protein in methicillin resistant Staphylococcus aureus. So this is another clue. MRSA, methicillin resistant Staphylococcus aureus, MRSA, is a part of this game. So this gene, what we were looking at, this sequence, is actually causing Staph aureus to be resistant to methicillin, to an antibiotic, because the gene binds to antibiotic. This gene that is now a protein binds to the antibiotic and causes it to be resistant. So again, lots of detective work here, but it's a lot of fun. And again, we solved another puzzle. This sequence, this unknown sequence, we didn't know what it was, is actually, to answer the questions, the pathogen is MRSA, methicillin resistant Staphylococcus aureus. Also, the gene is MAC-A that binds to an antibiotic that makes it ineffective. The gene binds to, the gene is translated into a protein that binds to the antibiotic and makes it resistant. And now we're gonna do some more activities with this. Again, detectives, well done.